Welcome to the fascinating world of genomic data analysis. In this video, we'll embark on a journey to understand how scientists decode the language of life hidden within our DNA. Genomics is the study of an organism's entire genetic material, and analyzing this data helps us uncover the secrets of health, disease, and evolution. From the basics of DNA structure to the advanced computational tools used today, we'll break down complex concepts into easy-to-understand ideas. Let's begin by exploring what makes genomic data unique and why its analysis is so crucial in modern science. Genomic data refers to the complete set of DNA sequences in an organism. This includes not just the genes that code for proteins, but also non-coding regions that play regulatory roles. With the advent of high-throughput sequencing technologies, scientists can now generate massive amounts of genomic data quickly and affordably. This data is stored digitally, often requiring specialized databases and software for management. Understanding what constitutes genomic data is the first step in appreciating the complexity and potential of genomic research. The heart of genomic data is DNA, the molecule that carries genetic instructions. DNA is composed of four chemical bases: adenine, thymine, cytosine, and guanine, arranged in a double helix. The sequence of these bases determines the genetic information unique to each organism. Understanding DNA's structure is essential for interpreting genomic data, as even small changes in the sequence can have significant biological effects. This foundational knowledge sets the stage for more advanced analysis techniques. Modern genomic analysis relies on advanced sequencing technologies. These methods, such as next-generation sequencing, allow scientists to read millions of DNA fragments simultaneously. The resulting data is vast and complex, requiring powerful computers to process. Sequencing technologies have revolutionized genomics, making it possible to study entire genomes in a matter of days. This rapid progress has opened new doors in medicine, agriculture, and evolutionary biology. The explosion of genomic data storage has become a major challenge. Each human genome generates hundreds of gigabytes of raw data. Storing, managing, and retrieving this information requires robust databases and secure cloud solutions. Scientists must also ensure data privacy, especially when dealing with sensitive human genetic information. Efficient storage solutions are critical for enabling large-scale genomic studies and collaborations across the globe. Before analysis can begin, genomic data must undergo rigorous quality control. This process involves checking for errors, contamination, and incomplete sequences. High-quality data ensures that subsequent analyses are accurate and reliable. Scientists use specialized software to filter out low-quality reads and correct sequencing mistakes. Quality control is a crucial step that underpins every successful genomic study. Once high-quality data is obtained, the next step is genome assembly. This process involves piecing together short DNA fragments into longer sequences that represent the entire genome. Genome assembly is like solving a massive puzzle where each piece must fit perfectly. Advanced algorithms and computational tools help scientists reconstruct the genome, providing a foundation for further analysis and discovery. After assembly, scientists annotate the genome to identify genes and other functional elements. Annotation involves predicting the locations of genes, regulatory regions, and non-coding RNAs. This step transforms raw sequence data into meaningful biological information. Accurate annotation is essential for understanding how genes function and interact within the genome. One of the key goals in genomic analysis is to identify genetic variants, differences in DNA sequences among individuals. Variant calling uses computational methods to detect single nucleotide polymorphisms, SNPs, insertions, deletions, and other changes. These variants can influence traits, disease susceptibility, and drug response. Accurate variant calling is vital for personalized medicine and population genetics. Functional genomics explores how genes and genetic variants affect biological processes. By integrating data from gene expression, protein interactions, and regulatory networks, scientists can uncover the roles of specific genes. This field bridges the gap between raw genomic data and real-world biological function, providing insights into health and disease mechanisms. Transcriptomics is the study of RNA transcripts produced by the genome. By analyzing which genes are active in different cells and conditions, scientists gain a dynamic view of gene expression. This information helps reveal how genes are regulated and how they respond to environmental changes. Transcriptomic data is essential for understanding complex biological systems. Epigenomics examines chemical modifications to DNA and histones that influence gene activity without changing the DNA sequence. These modifications can be affected by environment, lifestyle, and disease. Epigenomic data adds another layer of complexity to genomic analysis, helping scientists understand how genes are turned on or off in different contexts. Proteomics is the large-scale study of proteins, the functional products of genes. By analyzing protein abundance, structure, and interactions, scientists can link genomic data to cellular function. Proteomic data complements genomic analysis, providing a more complete picture of biological systems and disease mechanisms. Metagenomics involves analyzing genetic material from entire communities of organisms, such as microbes in the human gut. This approach reveals the diversity and functions of microbial populations, which play crucial roles in health and the environment. Metagenomic data analysis helps scientists understand complex ecosystems and their impact on human biology. Bioinformatics is the application of computational tools to analyze biological data. In genomics, bioinformatics software helps process, visualize, and interpret vast datasets. Popular tools include sequence aligners, genome browsers, and variant callers. Mastery of bioinformatics is essential for anyone working in genomic data analysis today. Visualizing genomic data is key to understanding patterns and relationships. Scientists use graphs, heat maps, and genome browsers to explore data interactively. Effective visualization makes complex results accessible and helps communicate findings to a broader audience. It's an indispensable part of the genomic analysis workflow. Statistical methods are fundamental in genomic data analysis. Scientists use statistics to identify significant patterns, associations, and differences within large datasets. Techniques like regression, clustering, and principal component analysis help extract meaningful insights from complex data. Robust statistical analysis ensures the validity of research findings. Machine learning is transforming genomic data analysis. Algorithms can classify samples, predict disease risk, and uncover hidden patterns in massive datasets. By training models on genomic data, scientists can make accurate predictions and automate complex analyses. Machine learning is paving the way for new discoveries in genomics. Population genomics studies genetic variation across groups of individuals. By comparing genomes, scientists can trace ancestry, migration, and evolutionary history. Population-level data also helps identify genetic factors that contribute to disease and adaptation. This field provides a broader context for interpreting individual genomic data. Comparative genomics involves analyzing the genomes of different species to identify similarities and differences. This approach sheds light on evolutionary relationships and the conservation of important genes. By comparing genomes, scientists can discover new functions and understand how genetic changes drive diversity in the natural world. Genomic data analysis is revolutionizing healthcare through personalized medicine. By understanding an individual's genetic makeup, doctors can tailor treatments to maximize effectiveness and minimize side effects. Genomic insights are leading to breakthroughs in cancer therapy, rare disease diagnosis, and drug development. Pharmacogenomics studies how genetic differences affect drug response. By analyzing genomic data, scientists can predict which medications will work best for each patient. This approach reduces adverse reactions and improves treatment outcomes, making medicine safer and more effective for everyone. Cancer genomics focuses on the genetic changes that drive tumor development. By sequencing cancer genomes, scientists can identify mutations, track disease progression, and develop targeted therapies. Genomic analysis is transforming cancer diagnosis and treatment, offering hope for more effective and personalized care. Many rare diseases are caused by genetic mutations. Genomic data analysis helps identify these mutations, enabling accurate diagnosis and potential treatment options. Advances in sequencing technology have made it possible to solve medical mysteries that once seemed unsolvable, bringing hope to patients and families. Genomic data analysis raises important ethical questions. Issues like privacy, consent, and data sharing must be carefully managed to protect individuals' rights. Scientists and policymakers work together to establish guidelines that balance research progress with respect for personal privacy and autonomy. Protecting genomic data from unauthorized access is crucial. Security measures include encryption, secure storage, and controlled access. As genomic databases grow, maintaining robust security protocols becomes even more important to prevent data breaches and misuse. Collaboration is key in genomics. Sharing data across institutions accelerates discovery and enables large-scale studies. However, data sharing must be balanced with privacy concerns and ethical considerations. International initiatives are working to create frameworks for responsible data sharing in genomics. Open-source software and data are driving innovation in genomics. By making tools and datasets freely available, the scientific community can build on each other's work and accelerate progress. Open-source initiatives foster transparency, reproducibility, and global collaboration. Cloud computing has revolutionized genomic data analysis by providing scalable storage and computational resources. Researchers can now analyze massive datasets without the need for expensive local infrastructure. Cloud platforms also facilitate collaboration and data sharing across the globe. Genomic data is a prime example of big data, with challenges in storage, processing, and interpretation. Handling such large and complex datasets requires innovative solutions in data science and engineering. Overcoming these challenges is essential for unlocking the full potential of genomic research. Artificial intelligence (AI) is making waves in genomic data analysis. AI algorithms can identify patterns, predict outcomes, and even suggest new research directions. By integrating AI with genomics, scientists are accelerating discoveries and opening new frontiers in biology and medicine. Clinical genomics applies genomic data analysis directly to patient care. By integrating genetic information into medical records, doctors can make more informed decisions about diagnosis, treatment, and prevention. Clinical genomics is paving the way for a new era of precision medicine. Agrogenomics uses genomic data analysis to improve crops and livestock. By identifying beneficial genetic traits, scientists can breed plants and animals that are more productive, resilient, and nutritious. Genomic insights are helping to ensure food security and sustainable agriculture for the future. Environmental genomics studies the genetic makeup of organisms in their natural habitats. By analyzing environmental DNA, scientists can monitor biodiversity, track invasive species, and assess ecosystem health. This field provides powerful tools for conservation and environmental management. Forensic genomics applies genomic data analysis to criminal investigations and legal cases. By analyzing DNA evidence, forensic scientists can identify suspects, exonerate the innocent, and solve cold cases. Genomic techniques are making forensic science more accurate and reliable than ever before. As genomic data analysis becomes more widespread, education and training are essential. Universities and online platforms offer courses in genomics, bioinformatics, and data science. Building expertise in these areas prepares the next generation of scientists and healthcare professionals for the genomic era. Public understanding of genomics is vital for informed decision-making. Outreach programs, educational resources, and media coverage help demystify genomic science. By increasing genomic literacy, society can better engage with the ethical, legal, and social implications of genomic research. Future of genomic data analysis is bright. Advances in technology, data science, and artificial intelligence will continue to drive discoveries. As costs decrease and accessibility increases, genomic analysis will become an integral part of healthcare, agriculture, and environmental management worldwide. Despite remarkable progress, challenges remain in genomic data analysis. Issues like data privacy, interpretation complexity, and equitable access must be addressed. Ongoing research and collaboration are essential to overcome these hurdles and ensure that genomic advances benefit everyone. International collaborations are driving progress in genomics. Projects like the Human Genome Project and the Earth Biogenome Project bring together scientists from around the world to map and analyze genetic diversity. These initiatives are expanding our understanding of life on Earth and fostering global cooperation. Citizen science projects invite the public to participate in genomic research. By contributing data or analyzing results, non-scientists can help advance our understanding of genetics. These initiatives promote engagement, education, and a sense of shared discovery. Governments and organizations are developing regulatory frameworks to guide genomic research and data use. These frameworks address issues like consent, privacy, and data sharing. Clear regulations help ensure that genomic advances are used responsibly and ethically. Standardizing genomic data formats and analysis methods is essential for reproducibility and collaboration. International organizations are working to develop common standards that facilitate data sharing and integration. These efforts make it easier for scientists to compare results and build on each other's work. Genomic data holds immense power to transform science, medicine, and society. By unlocking the secrets of our DNA, we can improve health, understand evolution, and protect our planet. The journey of genomic data analysis is just beginning, with endless possibilities ahead. As we conclude our exploration of genomic data analysis, it's clear that this field is reshaping our understanding of life. From decoding DNA to applying insights in medicine and beyond, genomics is at the forefront of scientific innovation. Continued advances in technology, collaboration, and education will ensure that genomic data analysis remains a driving force for discovery and progress in the years to come.